Hey guys, I'm going to be talking about DNA replication. So, when a cell divides in a process called mitosis, a copy of the old cell's DNA has to be put into the new cell. To do this, DNA makes a copy of itself through a process called replication. So, DNA replication starts with the enzyme helicase unwinding the DNA, forming what's called an origin. This is where the single strands are exposed. Inside the nucleus, there are these enzymes called endonucleases. And these are normally, a good thing. They're protective. They protect us from foreign DNA. However, when our single strands are exposed, they become quite dangerous. To prevent damage to the DNA, single-strand binding proteins attach to the exposed single strands. Once the strands are safe, replication can really get started because endonucleases are no longer a problem. There's one enzyme that does a lot of the work when it comes to DNA replication, and this enzyme is DNA polymerase. DNA polymerase is an enzyme capable of synthesizing new DNA strands. And because DNA strands go in 5 to 3 direction, DNA polymerase has to go in a 3 to 5 direction. The polymerase will go down the line, just matching each base with its little partner, A to T, T to A, C to G, and G to C. And when a match is made, a bit of the deoxyribonucleotide residue will be added onto the strand, and the polymerase will just continue on. Eventually, the entire strand is replicated this way. However, the strands that have been made have a lot of breaks in them, from where another origin was started further along the strand of DNA. Ligase enzymes will seal these breaks by forming the necessary phosphoester bonds. Polymerase, our favorite enzyme, will then go along our patched strand and make sure that there aren't any imperfections. If it should happen to find one, it'll just clip it off and they can try again. Usually this isn't the case. An error only really occurs once every 10,000 times. Finally, helicase enzyme returns to zip the strands back together. Whoop. Once everything is said and done, and our helix shapes have been reformed, we are left with 2 complete DNA strands, both half old and half new. So that's DNA replication in a nutshell. I hope you enjoyed, and I hope that was educational. Thank you for watching.